It is my pleasure to welcome our speaker for today. Martina Schifferer joins us, and she will be telling us about neuropathology on tape, a volume EM-based application. Martina is the associate investigator at the Electron Microscopy Hub at the DZNE, the German Center for Neurodegenerative Diseases. Martina studied molecular biotechnology at the Technical University in Munich and got her PhD in biology at the Max Planck Institute for Euro-BioImaging. Following a postdoc at EMBL in Heidelberg, she's been at the Electron Microscopy Hub in Munich since 2017. And we're really looking forward to hearing from Martina today on her work on using volume EM to understand neuropathology. Please go ahead, Martina.
Thank you very much, Johanna, for the kind introduction and also for inviting me to present our work here today, and also thanks to the volume EM community. So what I would like to tell you today is a bit the applications, but also the methodology that we are using, the so-called ATUM method, which is a specific kind of volume EM method and a specific type of array tomography. And I will show you several examples that we apply in our EM hub in Munich. So I will first start with the neurobiological questions that we are facing in our core facility, and then talk about the EM techniques, our EM techniques that we are applying, and then it hopefully becomes a bit clear that for some applications, ATUM is a very good method. Then I will quickly explain the workflow for you to get an idea about the technical details. I show you some applications, and at the end, I will also talk about some technical variations of the method that we are working on at the moment. So please also feel free to interrupt me and ask any questions in between. The Neurosynergy EM hub or nanoscale hub is located in the south of Munich, and that's actually here our building at DZNE. It's the German Center of Neurodegenerative Diseases. We are here situated in the second floor, not the best location for an EM facility. And we have a room pit temperature electron microscopy. Here's one of our team members, Georg, with a TEM, and here's Connie with our size crossbeam. And we are embedded in a research consortium, so the German Excellence Cluster, it's called Synergy, and the topic is neurodegeneration and neurodegenerative diseases as one of the applications, and that concerns mostly axonal damage or neurodegeneration in general. Then we have another topic, which is neuroinflammation, so all kinds of immunological questions. And then a third part is the gliovascular dysfunction. And we are normally working with mouse models, but we also have some zebrafish and some human biopsies, as well as some recent animals. So we have also mini pig models, but we are mostly working in mouse, and that's also what I'm showing you today. And regarding the model systems, we are then using mostly tissue, so CNS or penis tissue, and sometimes also in vitro systems or organoids. And just to give you an idea how this technical application is embedded within this consortium, so we have several hubs, several technology hubs that offer different kinds of imaging or other technical modalities. And that's especially important for neurobiological research because, as you can see, the scale ranges, so it extends over 8 powers of 10. And that explains also why these different methods are really needed to visualize these different complexity levels of a brain. And here I would like to show you one example of a human biopsy and how at the end our nanoscale hub is embedded in this whole procedure of analyzing the sample. So here we have an MRI, which is was generated by the macroscale hub. Then a lot is done in the microscale / light microscopy hub regarding histology. And then we have been looking in this case in this Borna virus autopsy into replication centers of a virus. And this is something, of course, that you can only resolve with electron microscopy. We are a room temperature electron microscopy facility, and these are our 3 microscopes. And this is just to put somehow into scale what we are doing with these different kinds of microscopes. With the SEMs, we are mostly looking at large field of views. For example, here in this cross-section of a mouse optic nerve. And then with the TEM, we can really look down to the single layers of myelin sheath of the axon. And with both microscopes, we can look at something in between. So then about the techniques that we are applying. So far, I only talked basically about 2-dimensional EM. But in some cases, we really need the 3-dimensional information. And I guess many of you know what 3-dimensional imaging is good for, but here's just specific example from one of our collaboration projects. So the initial question was, so the collaboration partners, they had a mouse model for neuromyelitis optica. That's a demyelinating disease, so the myelin sheath in the spinal cord and also in the optic nerve of the mouse is degenerated, and they observed in their 2-photon imaging this kind of beading. So the axons were thicker at some sides along the length of the axon, and then we looked at cross sections of these, and then of course, 2-dimensional imaging doesn't help you too much because there could be 2 different scenarios. One is that this is a site of a bigger axon, so an axon with a larger diameter, or if this could be also a swollen axon. And we were really interested in the morphology of this swelling. And in our facility, we have 2 different kinds of 3-dimensional imaging, volume EM imaging. So the ATUM method, which is called automated tape collecting ultramicrotomy, and this was invented in Jeff Lichtman's lab. And the other technique we are using is the focused ion beam SEM. And these are now just 2 of the available methods, but they are also representatives of basically the array tomography approach versus the block phase imaging. And the huge advantage of the focused ion beam SEM, as you most of you know, is that the set resolution is not limited by the physical sectioning thickness of the thickness of the section, so by physical sectioning, but the beam milling allows voxel resolution of 5 by 5 by 5 or even lower. And this is really a disadvantage of the ATUM method, that we are limited here to like I say, something like 50, but also down to 30 nanometers. But that's usually the range, and we cannot go down to 5 nanometers just because of the sectioning. But still, we can use this method. For example, here, when we want to look at cell-cell contact. So here we have different glial cells wrapping around a blood vessel. As I told you, we were interested in these cardiovascular diseases like stroke and so on. And for these kind of questions, this is quite a good method that can be used despite this disadvantage. Then another application of the FIB SEM was a so-called double myelination model. So you see here in blue, this is the axon, and then we have a mutant, so a double mutant actually, of 2 adhesion proteins, and that mess up basically with the wrapping of the myelin around the axon, and that results in 2 myelin sheaths that wrap around the axon, the green and the pink one. And we wanted to understand the geometry of this wrapping, and for this, these isotropic voxels were especially important. And so now I'd like to show you like the different steps that have to be taken for the ATUM method. So first, we serial section with an ultramicrotome, and I will go into the details of the steps later, but just for you to know that these sections are then taken up by a plastic tape, and then the plastic tape is glued onto a silicon wafer, and each section is then imaged sequentially and reconstructed, and then we can segment different objects, for example. And this is now this example of this neuromyelitis optica, or NMO mouse model, where you can here now see on the left side, basically the main advantage of the ATUM method, that we can image the same sample or same region even in the sample with different resolution schemes. And usually we then, so this is called hierarchical imaging, where, for example, here we can scan the whole cross-section of the spinal cord and then zoom into the region that was affected by this treatment to generate this mouse model, where you can see that these axons are really disrupted. And then we can really only reconstruct from this part of the tissue. And, oh, that was a bit too fast. Yeah, and then these are the different sections generated, and the model here of two really swollen axons, one unmyelinated and one myelinated axons. And then we can zoom into these regions and can really identify the outer structure of these axons. So this was basically what the volume EM is about in general, but there is one specific advantage of the ATUM method that concerns targeting neuropathologies, and that's why it's so interesting for many of our applications. So remember, I told you at the beginning that so the overall size of the brain is quite big, and then we are looking at single cells or cell-cell contacts, and they can be rare, and they can also be distributed randomly in the brain. And that's, for example, for these mice models, sometimes it's really from mouse to mouse a different location. And therefore, this is often a needle in the haystack problem. And so this sketch should show you how we can approach this problem with the two different kinds of volume EM techniques. So ATUM, as I said, can in this case now also be replaced by array tomography. So any kind of conservation of sections on a support, and the FIB-SEM as typical block phase method. So for FIB-SEM, usually the targeting has to be really precise because we have only one shot, so we can only target this region of interest once, or we can try it several times, but that's very time consuming. And usually micro-CT or any other additional experiment is needed to really get to that region of interest. And I think regarding the targeting of regions, I would say that array tomography in general is really very forgiving, but because you can first image at low resolution, very large areas, and then slowly zoom into your region of interest. And you can, as I said, that's very much easier screening process. And it gives you also the advantage to look at different sites with different resolutions also at a later time point. And I mentioned that ATUM is basically a variant of the array tomography technique. So everything I'm saying now about the advantages kind of also holds true for this, what I like in comparison to ATUM call now solid support array tomography. I just want to point out that the sectioning and collection is, in my opinion at least, a bit easier in the ATUM method. Because you don't have to generate these ribbons, which generally takes a bit of training. And we can also generate larger sections, which helps then in this case of searching for certain pathological sites. The disadvantage is that you collect less sections per area, but not really less area of tissue. So bigger sections, but less. And like technically, Both techniques are not very expensive compared to the block phase techniques, because in the AT you need just some support and maybe a special knife and some ribbon collection device, and at the end for the microscope, a serial section acquisition software. Everything that's now depicted in white is considered to be part of like a standard EM facility. And for the ATUM, it's of course the tape, then the tape collector that can be installed next to a microtome. Then what is recommended is a plasma discharger, but I come to that for the tape. And then wafer mounting material, but this is all not very expensive. Okay, so I was mentioning a bit the workflow, but now I'd like to go a bit into the details. So here is something about the overall workflow and the timing. So we start with sample preparation, that is normal rotor protocol. Then we go to serial sectioning and collection. And then we basically have our sections on the tape, and then we have to glue these strips of tape onto wafers, do the imaging and the image analysis. And this is also to point out that at a quite early stage in the whole process, the axial resolution is determined, of course, by the sectioning, but this can be quite variable, and I will come to that a bit later. So this can be in between 50 nanometers and 5 micrometers, but this decision has to be made quite early. But then these green arrows should show you that with this hierarchical imaging, we can at a later time point then determine the lateral resolution. Okay, so we start out with a sample processing, and so usually we have vibrotome sections or blocks of tissue from brain or from spinal cord, and for this we use really standard rotor protocols like the Alisman or the Euro protocol, but in our case we see that the lead step can be omitted and we leave out the lead step. So that's our typical contrasting protocol. Then another option is either for blocks you already have that you are contrasted, where you didn't do a rotor contrasting, but where you only have osmium and UA. The wafers can then be post-stained. So once the sections are collected on the wafer, you can do some post-staining, and that's, I think, quite nice, especially if you have very precious, for example, human biopsies or other very precious samples that are already contrasted, and you can still then do volume EM on them if you do this post-staining. And then there are, for the embedding, there's one trick that we have used in the past, but not that often. So one of the main problems of array tomography, or of ATUM especially, is that the tissue can have folds. And to overcome this, it's very one way is to embed the tissue in a kind of support tissue, meaning in a non-important tissue that you position around your object of interest. And then we use LX112, which is an epoxy araldite resin, and this seems to be a very well suited for the ATUM application in general. So that's our standard resin. For the trimming, we started out with trimming at 90-degree angles, but because of course then every section has the exactly same shape, but neither for mapping nor for sectioning itself it's really necessary. So we now use our trimming machine and usually have an angle between 45 and 60 degrees. And as I said before, you can use this so-called support tissue that is lighter gray here. That's better than having a lot of resin around your tissue of interest. And that's especially true if you have a very small sample. And then one specific thing you have to remind for the ATUM technique is that the angle of the tape cannot be changed. So you have to trim in a way that the block is really parallel to the tape. So so don't introduce any angle there. That implies also for, like, you have to take care of that in the embedding already. So then one of the main parts of the ATUM technique, and also one of the main challenges, is the tape. So the support material. So it's a plastic tape, and usually Kapton is used. But one characteristic of this tape is that it has to be conductive. So Jeff Lichtman's lab invented carbon-coated Kapton. So they have a coating machine for this. And then you can use SE and BSD detection. And then there is the carbon nanotube or a CNT tape available from, for example, here in Germany, Science Services, which is also conductive and was introduced by the Kubota lab. And we are also using that regularly. But here is an example now of this is an optic nerve sample, 30 nanometers in thickness. And you can see that for the CNT tape, we get some background pattern. And another disadvantage of the CNT tape, at least in our hands, is that so these are now binocular images of the sections on the tape, and you can see that you cannot see it very well. And in contrast, the carbon-coated Kapton, you can even distinguish the different thicknesses of the sections, which is very nice. The only problem is that at the moment, the CNT tape is not even commercially available, and the carbon-coated Kapton is not available at all. There is still the possibility to carbon coat on top of the Kapton. And then in order to avoid faults, it's recommended that the Kapton tape is also plasma discharge, and this shows the tape before and after discharge. And this is also a custom built plasma discharge that we use in the lab. And then this is how our microtome looks like, and we put it into an enclosure, especially for longer runs, that has a lot of advantages. And yeah, here you can see the sample and the water bath, and here is basically the nose of this tape collector. And this is a side view of the tape collector, and this is how you basically introduce the tape into this tape collector. And then at the end, if you have successfully collected, then the tape is glued onto the silicon wave, and you can see that we have another layer of carbon adhesive tape, and then we have to ground the wafer also with carbon adhesive strips in addition. And then, by the way, this is also why it's called automated. So it's a very time-consuming process, especially if you have a lot of wafers. And then what we found is very nice is before we introduce our wafers into the microscope, we have a wafer drying station that was built by Binder Labortechnik and Science Services. And it reaches vacuum values of up to 10 to the power of -7. And it really helps to keep our microscopes clean. And yeah, and then in addition, we have this wafer airlock for the size microscope. And so I told you at the beginning that we have two different SEMs. So this is now the crossbeam that we're using, and we are using the Atlas software for this. But we have also another an Apriu system with a map software that is a bit that is different to as it's also an array tomography software, but it's not really built for ATUM, and has some advantages and disadvantages regarding in comparison to the Atlas 5. But now I'm just showing you our general workflow. So this is the wafer in the chamber, and then we do the mapping by really mapping the whole wafer at a very low resolution. And then you can detect here the single sections, and you see that the collection was not perfect, but this is basically a normal life. And then this automatic detection of the sections didn't really work here, and we have to correct for this. And then we can select our region of interest that you can now better see here in this image. that we then choose for medium and and here also high resolution scan. And in this case, this is an Alzheimer disease plaque. It's a mouse model for Alzheimer's disease. And this is like a typical scale of our project. So we have something like 4 wafers, 250 sections each, so 1,000 wafers. And then we first screen these 4 wafers, and in this case, one plaque has something like 50 to 80 micrometers in diameter, and we look for one plaque that really is covered by this thickness, and then we can do registration of this plaque. And here again, you see this hierarchical imaging. Like first you look at the cortex, then you screen into one of these plaques, and then here this is a microglial cell with extensive ER that is contacting the amyloid fibrils. And then this is the reconstruction, and on the left you can see the sections, and you can also see that there are sometimes some folds popping up, which is of course a problem in image analysis. Yeah, and this is just the whole model reconstructed. And we can also then zoom into certain regions. For example, here we see that the A beta is really surrounds these blood vessels here in red, and the microglia try to keep the amyloid kind of shielded to prevent spreading. And this is another application where we were specifically looking at iron distribution by a histological stain. So here, now in gray, you see the amyloid beta, then in white, these are the microglia surrounding this plaque, in red, you see the blood vessels, and in blue are the parts of the microglia that took up the iron, and you can also see the iron deposits on the blood vessels. Yeah, and then not to forget about the problems. So we have this fold formation that can have different reasons, and I won't go into details. But the main messages are that the tape discharge is very important for the collection folds, and then the support tissue can also be advantages. And then it's, of course, in this case, always better to have less heavy metals. But then on the other hand, and this is something that keeps us very busy, is that a high heavy metal load is, of course, good, especially if you want to do a repetitive imaging. Okay, so now I'd like to show you some more applications. And for this, I would like to basically point out like the 3 main challenges of electron microscopy. One is that it's not dynamic because we are looking at fixed states. The other is that it's not specific because of the heavy metal load, and the other one is a limited field of view. And all of you know that one of the ways to overcome this is to do correlative imaging. And that's one of the applications of the ATUM techniques. So here we collaborated with a group that is interested in stroke, and they induce the stroke with a magnetic nanoparticle. So this is now this cranial window, and you see that before and after these magnetic particles that were previously injected, they are not visible here, and upon application of the magnet, then you can really induce stroke in this area. And we also use fluorescent nanoparticles to monitor this, and then we can look at different time points. So here, time point 0 and 60 minutes at the very same side of the vasculature. And here the arrow points to vascular occlusions occurring at different time points. And then the nice thing about this method is that these magnetic particles are electron dense, so we can actually use these particles to do the correlation. So we can overlay here the light microscopy and the SEM images and find the same region back. And this is now what is shown on the left side, reconstructed. And here we can really reconstruct all the different cell types that are in this region and understand how these clots in this stroke were composed. And we also found back this specific region where we can then analyze the ultrastructure of this so-called primary and secondary clot, and that's very important in stroke research. Another example of correlation is shown here in mouse neuromuscular junction. So this is actually the nerve that is innervating the muscle. And BTX is bungarotoxin, and lyso-T is a lyso-tracker. And so this is in collaboration with a group of Thomas Misgeld, where we were looking with the PhD student working on this project at trafficking of mitochondria in and out of this neuromuscular junction. And you can see here a larger field of view with the neuromuscular junction highlighted here with this arrow. And by using this technique called near-infrared branding, where you really use high laser power, we then labeled this area. Here you can actually see the muscle fibers. And we then reconstructed this area. So this is the very same neuromuscular junction as reconstructed from the electron microscopy. And we found then this specific site that was imaged with light microscopy, and we identified this as mitophagy. So these used to be mitochondria and are now kind of lysosomal structures and are degraded in that specific site of the neuromuscular junction. So we can now really use this ATUM method to screen for the area of interest and then acquire this certain structures with high resolution. And the last point is about the technical developments. So as I told you, that the correlative imaging is one way to overcome these limits, but we thought that this ATUM method could be also very well suited to overcome the limitation of this low, small field of view. And that's now showing that with the different section thickness, you can already look at different kind of biological structures. So as you all know, this was invented for connectomics, where it was really important to have a 30 nanometer very thin section to really map and not miss any of the synapses. But in our applications, usually 50 or 50 up to 200 nanometers are very suitable for many applications. So 50 we usually use for organelles and cell contacts. But then here, for example, we reconstructed the whole model, but then at the end we were only interested in one two-dimensional image in this case of this degenerating axon. And the advantage, of course, is then that the screening speed is much higher. And we came up with this basically idea that I showed you at the beginning, which is actually quite simple, that you have the ATUM for the screening and as a second step, the image acquisition. And then if you have thicker sections, like 5 micrometer thick semistic sections, you can screen much faster. And at the end, choose your object of interest and do FIB-SEM on that. And for this, we had to develop heated knife, because we had a problem that at room temperature we got a lot of cracks in these thicker sections. And together with Helmut Gnägi, he helped us to develop this knife, which is actually a very simple version of the hot knife method, and which works quite well in our hands. And then we so the surface is much smoother than at room temperature. And with this, we can then look at semi-thick sections that allow us to also do light microscopy or low resolution SEM to search for a structure of interest, and then do FIB-SEM on that structure. Okay, and then the last few minutes, I'd like to talk about another parameter that is really limited in EM analysis, which is a molecular identity. And of course, CLAM is very useful for that, but usually in CLAM, there are only one or a few proteins or cell types labeled. That's why we teamed up with a spatial transcriptomics group in basically also within the Synergy cluster. And we had the idea to combine EM with spatial transcriptomics, because there we can really look at thousands of transcripts. And of course, for them, it was very interesting to have some imaging technique that would like highlight or visualize the relevance of the differential transcription. But the sample preparation for both techniques is very different. And that's why we thought it would be best to do a cryosectioning and then only collect the adjacent sections onto glass slides. So that means we are only investigating adjacent sections, not the very same section with both techniques. And the model that we used was one of our usual models, the white matter injury. That means we inject some kind of soap that dissolves the myelin sheath and then demyelinates the axon. And then upon demyelination, there is an immune reaction with microglial cells that are then located in this lesion site. And so one of the challenges then, because the spatial transcriptomic sections are very precious, we want to keep as much of the area as possible. And that's one of our giant samples with almost 8 millimeter diameter. At the end, we were then only interested in part of this, but it was important for us to keep these sections from the beginning. And then both with spatial transcriptomics and EM, we classified the microglia, so this special cells that are migrating into this lesion, and we could, with spatial transcriptomics, identify certain classes of microglia, including, for example, foam cells that have a lot of lipid droplets. And these lipid droplets we can then also see in the EM, which so the ultrastructural quality of this EM is, of course, not perfect, because we have to really compromise with the sample preparation of the spatial transcriptomics. But we could identify differences, and we are at the moment really looking at, we are segmenting all the different organelles there and then comparing the organelle content and different other features like the nuclear heterochromatin pattern, and try to distinguish the different classes of microglia. And yeah, with this, I would like to thank the people that actually did all the work. So that's my team, Connie and Georg, which really are amazing team members. And this was also done with the help of some students, especially the segmentation was done by Hanyi, Lucia, and Anya. The hub is coordinated by Martin, Thomas, and Mika. And I'd like to especially thank Richard Chalek and Mark Terasaki for introducing me into the ATUM method, and Helmut Gnegi for all the help and like the technical support over the years. And I also would like to thank all the collaborators that shared all their expertise on the scientific part of the project. So with this, I'd like to end, and I'm happy to take questions.
Thank you, Martina. That was a fantastic talk, really interesting and very impressive work. Are there any questions from the audience? You can raise your hand or just unmute the microphone and ask the question, or put it in the chat if you prefer. Maybe about giving people a chance to think about questions, I could ask the first one. You mentioned that to prevent these folds, you're embedding sometimes the tissue in support tissue. Maybe this is just something I'm not familiar with this at all. Can you talk a little bit about like what kind of support tissue that is, and how you then separate the real tissue of interest from the surrounding?
Yeah, that's actually, so the separation is not really a problem, because you always have basically a gap between your real tissue and the tissue of interest. And once you section your tissue, you really see what's your tissue of interest. And usually, so yeah, we just have a lot of fixed brain where we only take parts of the brain for our samples, and yeah, and then we usually take cortex tissue because it's it can be sectioned very well. So in compared to white matter regions with a lot of myelin that's quite problematic for the sectioning, and then we just put this cortex tissue around it. But as I said, it's we don't use it so often. But for example, in this spinal cord sample that I showed you, where really the region of interest is at the site between the empty resin and the tissue, so we really use this to decrease this fold formation, because especially in sites between empty resin and tissue, this folds form more often. Yeah, and in this case, for example, what we just did is we put two sections next to it that were both of interest next to each other, face to face, and then we had the region of interest in the middle. It's just to avoid this empty resin to tissue transition.
Okay, thank you. That makes a lot of sense. We have a question from Jens in the chat. Jens, do you want to ask the question yourself?
Yes, absolutely. Thank you for a very nice talk. We were interested in this drying chamber that you used to dry the wafers prior to imaging on the SEM. And we were wondering, is it only working with vacuum, or does it have desiccators and the heat as well? And also, how long do you dry them before putting it in to avoid contaminations in the chamber?
Yeah, so thanks for the question. So this was actually especially built for these wafers, and it's also available from Science Services now. And it's equipped with a heating, but we usually don't use the heating. So because we had problems with the heating, that the tape comes off and you get really bubbles. So it was horrible. I think the company is working on it, but we also see that without the heating, it's working very well. And regarding the timing, I think I don't have the time. I don't have the graphs here now, but so usually we do that overnight. So we prepare the wafer, then we just put them in and then into the microscope, and that really is has a lot of advantages.
Thank you.
Okay, and there's a question from Susanna in the chat. I'll just ask that. She's asking whether you have some quality control measures across the process, so like controlling section thickness consistency.
So we don't have that, but I think it would be nice to have at least like also for the registration, it would be nice to have a measure for the actual thickness of the sections, yes. But no, we don't have it for the temperature and basically the airflow and so on. This is of course regulated in the room, and then we have this enclosure where all basically the environmental factors are minimized. But as I said in the beginning, we're also on the second floor, and there are also MRIs, and so there's a lot of different environmental factors that are still. I think that is one of the reasons why we can still not really reliably section down to 30 nanometers. At least, I mean, we do section 30 nanometers, but not thousands of sections, and this is, still a limitation.